Good afternoon, everyone. It's wonderful to have you joining us today for this masterclass on drug discovery bench to bedside. I'd like to begin by acknowledging the traditional owners and their custodianship of the many lands on which we meet today. On behalf of the University of Queensland, I pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. My name's Louise Hickson. I'm an Associate Dean in the Faculty of Health and Behavioural Sciences, which is where pharmacy is based. I'm delighted to be here this afternoon and to be your MC for this talk. And the format of today is that we'll have about 35 to 40 minutes of a presentation by Associate Professor Pete Cabot. And you can see the topic there on the screen. And after that, there'll be a great opportunity for you to ask any questions that you might have. I'll moderate those questions. So what I'm asking you to do is during Pete's presentation, if you could write any questions that you have in the chat, and I'll keep an eye on those and I'll put those to Pete at the end of his talk. So I'll remind you of that as we go along, but we think that's the best way to run this kind of question and answer format on Zoom. So without further ado, it is wonderful to introduce my colleague, Pete Cabot. Pete is an Associate Professor from UQ's School of Pharmacy, and like many of our staff, is a brilliant educator and researcher. He is the Director of the Master of Pharmaceutical Industry Practice, which he's going to talk to us about today. And as well as being a university academic, he has 20 years of experience in preclinical drug discovery, which is obviously central to the talk this afternoon. And he's worked with numerous pharmaceutical industry partners, such as companies that you would have heard of like Pfizer. So I'll now hand over to Pete. And remember, please put your questions in the chat.
Thank you, Louise. Thanks for the lovely start to the talk. Thank you everybody for coming along this evening. Or whatever time zone you're in, of course. Thank you for attending. What I'd like to do today is I guess it's a tale of two cities in some ways. First, I want to give you an insight into the drug discovery process and drug discovery life cycle. And we're going to start and really focus heavily on, I guess, an area of expertise for me, which is the preclinical drug discovery. So we're going to talk about where drugs come from. And then we're going to look at some of the examples of where we've created medicines, particularly out of UQ. And then that will take us pointedly through or segue nicely, hopefully, into the MPIP program. We'll talk about what that does in terms of learning around this drug life cycle process and its engagement in that course. So without further ado, let's get started. So I'll just minimize that. So, and of course, I'm a scientist, as Louise mentioned, and this, of course, is a photo pre-COVID, because we're not socially distancing in this photo, but this is me working with one of the students in the labs here at PACE. We're looking at novel painkillers and novel inflammatory targets in tropical rainforest plants and extracts, so which sort of lends into where we're headed with the talk next. So what brings us to talk about these concepts today? Well, really, it's probably the size and scope of what the market is for medical devices and pharmaceutical treatments. And so we see projections of enormous amounts of money where we're thinking the global market will reach something in the order of $3 trillion by 2025. It's an enormous amount of money. So the drivers for health and the drivers for this industry stem from probably aspects in society that we know that are changing and evolving, and the market has to meet the needs of these sorts of challenges. And so some of these challenges are things like the aging population, the increased prevalence of chronic illness, probably increased awareness as well in health, increasing healthcare costs, and of course, the onset of more personalised medicines and personalised therapies. as people take more medicines, we see lots more interaction. So that complexity means that we have to have new medicines in order to deal with these problems and issues. So that's probably an incentive for the rest of the talk, and probably the incentive for drug discovery as a whole. So in terms of a process, you can see up here across the screen is a pictorial of the process from discovery all the way through to the evolution of medicine. Excuse me. On the bottom scale, you can see large numbers of compounds or ideas and concepts coming in at one point and ending up with a single entity at the other. And up the top, you can see the enormous costs and the timelines that encountered in this drug discovery process. What I want to do next is focus on the one aspect down the front, which is kind of where I come from, and we'll sort of lend across to the rest as we walk through it before we talk about the MPIP. So drug discovery, we think of drug discovery as the white-coated scientists and that drug discovery process. And what we're actually trying to do in drug discovery is actually map out how the balance works. How do the chemicals work in our bodies? So here, this is obviously neurons connected together, and they're interacting in the graphics over here. And as a scientist, what we see and what we learn about, we end up with a picture of what this balance is for this neuronal firing and all the different types of sites that are involved in this process. In discovering that, we understand more about the nature of achieving balance and maybe when things change in illness. And therefore, we end up with a lot of drugs that are developed around, okay, we need to target this certain receptor or this receptor in order to bring balance back into shape, particularly when we lose that balance, say in an illness or an injury. So where do these drugs come from? Well, things have evolved, but some things stay the same. And of course, traditional sources are still a great source for medications that are on the market today. And this is probably one of the oldest and probably one of the still most prevalent used drugs. Now, I'm a pain and inflammation researcher, so I apologize immediately for, I'll have a tinge for going towards pain type medicines in terms of, because it's the kind of thing that excites me when I explore the concepts around the pain medication. So, and I guess in that I've given this away because this plant, Papaver somniferum, is historically known as one of the oldest cultivated crops in mankind. It dates back to, there's evidence back in Neolithic times of the cultivation and use of the opium poppy. And so, of course, the main constitutive ingredient that we use as a medicine today still is morphine. Morphine named from the god of sleep, Morpheus, by the Greeks. So it was a term that was applied because of the sleep-inducing aspects as well as the pain-relieving aspects. They believed that really the addiction wasn't understood, essentially, but the benefits were certainly very strong from taking the opium. And of course, we have a number of wars through history that were built around just a trade in this plant and its extracts. And so looking at traditional sources, these come from a number of nations, such as traditional bush medicines from the Australian Aborigines across a whole range of cultures through history and time. The thing with historical traditional sources is sometimes we lose a grip on understanding the seasonal effects or the regional effects and how those extracts might contain or behave in a person. That tends to be knowledge that has to be passed down through time. So drug discovery tends to move on. One of the parts where it did move on, or one of the concepts where it did move on, which is probably not a great concept, was glycoheroin. And of course, as you can see in the name, this is actually a combination of glycerin and heroin. Heroin was a modification, a very small modification, diacetyl morphine it is. So a very small modification to morphine itself. This was effective as acting as a cough suppressant. And of course, unfortunately, was actually promoted for use in children for a whole range of ailments. And in this case, sometimes mixed with cocaine as a soother, which I'm certain it did. However, the problem with this is this actually had high addiction potential and caused a major change in the use of heroin in a very short time of use, actually, in around about 20 years of use. Now, serendipity is one of the great advantages you can get sometimes. You're just hoping that in your scientific process of application to discovering a medicine that you also have the luck that comes along with it. And I think whenever we search up serendipity and drug discovery, we end up with penicillin as the key example. And so the story goes that Fleming went on vacation, which is always a good thing to do, but he left his Staph aureus culture dishes with incubating with Staph aureus across the plate. And when he returned, he noticed that, and so he was in the pursuit of a cure for bacterial infections. And when he returned, he noticed that there was a mold that had grown on the plate. Now, the scientist part of this is where, if I hark back to my days and sharing flats with colleagues in my early 20s, that was something disgusting that sat in the sink and you're trying to find who it owned to. In terms of what Fleming did, is he actually saw that, hang on, there's actually a ring of cell killing of the Staphylococcus around that mold, and hence the discovery of penicillin was made. It wasn't until about 10 years, I think it was 1943, before the Americans and English actually promoted it and got it out as a medicine. So it had some delay. It was because Fleming had a lot of trouble in isolating the compound itself. But that initial discovery was quite important. So scientifically developed based on disease, this is probably the greatest example of that discovery process. This is Banting and Best, and of course pictured here with Majori on the roof of the University of Toronto. And this is where Paul Langerhans actually found an islet cell group, the islets of Langerhans, and had implicated these pancreatic cell types in diabetes. And so Banting had read on this and had decided to go back and research the possibility that there was a direct link between something that was in the pancreas and the diabetes, because diabetes type 1, that insulin-dependent diabetes, had a terrible consequences for patients. A lot of patients in the early ages were in comas, and there was really no cure or treatment in order to control it. So the discovery of insulin was a very, very important discovery at the time, as you could well imagine. And of course, he scientifically applied the thought of, well, if I take the pancreas out of the dogs, we know it's been observed that they end up with something like a diabetes type effect. And then he injected back extracts of pancreatic tissue back into these dogs and found that their diabetic recovery was, and they were quite normal. So I think it was in a matter of a couple of years that they ended up in the first patient in a clinical trial, which was a 14-year-old boy, and numerous patients after that, where they actually could bring them out of coma with these pancreatic extracts injections. Of course, we don't use extracts anymore. We manufacture them in cell lines, but it's still the same principle. So the modelling of medicines, well, so then when we learn a lot more about structure-activity relationships in chemistry, and so I'm not going to do too much more of this. I know showing pictures of chemical structures is not a good idea, but what we do as scientists is we can see the pharmacophore in these, and so this area of this compound, which is called fentanyl, is consistent with this part of the nitrogen here and out to the ring structures, sorry, this ring structure up here. So although you can't see it, in two-dimensional, it's never clear, but in three dimensions, we can visualize these types of pharmacophoric structures. What this allowed us to do, in essence, was knowing these structures, we could actually build hundreds, if not thousands, of chemical mimics of these that still retained the pharmacophore. There's that pharmacophore I talked about before. Here's another one here. You can see this one's majorly modified. And this one here I've got upside down and back to front for some reason. But you can see it there and that. So that's as much chemistry as we're going to learn today. And hopefully that's enough for everybody. These are all fentanyl analogues. So part of that discovery process of building these compounds, they ended up with a whole range of different molecules. And then they go through the process of assessing them one by one or in small groups. Interestingly, you can't really tell anything from looking at these flat structures about their efficacy. And certainly 3 of these are potent analgesics used clinically today, very good painkillers. They still have addictive properties that wasn't removed, so it didn't solve this. One of these is actually really licensed more as a veterinary purpose. It's actually meant for sedating large animals. So that's carfentanil. And so it's actually that potent. It's actually 10,000 times more potent than morphine and 100 times more potent than fentanyl. So it's very potent. So if you can imagine a morphine tablet, looks something like a Panadol tablet, you'd have to cut it up into a 10,000th of that size in order to take it safely. So very, very dangerous. And so the type of animals that it's used for sedating is for treatment of animals like you can imagine, elephants and rhinos. Of course, it's also a weaponizable agent, which is another issue that occurs often with these types of chemicals. And there's a whole history of these with other examples with phosphate compounds as well. But this is the opioid one. And this is the analysis. This is a report on the analysis of clothing and urine from, if you remember, in 2002, there was a Russian special forces siege upon Chechen terrorists that had hostages in the Dubrovnik theatre, and they pumped in a mysterious gas mixture in through the ventilation system, and it resulted in a large number of deaths in the theatre. And so when the analysis was done on the clothing and the urine, they found carfentanil and another type of fentanyl analog, which was in this tissue. So I guess you can connect the dots, but certainly a very dangerous chemical. So how do we do it today? Well, this sort of thing that I do in the lab with me and my colleagues in the projects that we're working on with pharmaceutical companies right now at this very moment. And so we do it more with molecular modeling. So we're not building hundreds of thousands of chemicals. We're actually doing in silico analysis where we know what the structure is of the protein that we're targeting. So this is a delta opioid receptor over here. Remember I showed a 3-dimensional structure? Well, here's the 2-dimensional picture, and then here's how it looks in 3 dimensions. And what we simply do is put these two together using the software, and you end up with something like chicken wire down in a playpen in a kid's yard. But in reality that's in a kiddie center, sorry. In reality, that is showing the fit of this molecule into the receptor binding pocket of this receptor protein, so down inside that receptor. And that allows us to do in silico analysis of these binding associations. So it gives us an insight into how our compounds might behave, so we can manufacture them now in silico, assess them in silico, and that means the ones that we then make are more likely to be successful. So it saves a lot of resources, gives us a lot of extra time in terms of preparation. And this is what scientists in drug discovery are now doing all over the world, and pharmaceutical companies. Alongside this, we also do, and I routinely do these sorts of things, which is assessing using things like high throughput screening assays. And this might be where, and we haven't forgotten nature, we learn a lot from nature. So we go back to natural sources and look for activities. So, and Australia is particularly rich in this space because we have such a unique environment. If you look at areas where I've sourced extracts from as well, tropical North Queensland, where you have the Great Barrier Reef, where these friendly pho come from, and the rainforest as well up there. So you have a very aggressive environment there. So the biologists tell me where plants and animals are fighting to survive against each other in high density of each other. So they do things, they adapt to that environment. So they protect themselves, and they might protect themselves with an antibiotic. They might protect themselves with an antifungal to push away those that might infect them. And they might also have defenses or weapons, as these cone snails do, in order to catch their prey. As you can imagine, a cone snail can't move very fast, so it has a barb that releases a toxin. That's called these conotoxins. And these conotoxins, my colleagues at the IMB have explored the effects of these conotoxins as potential pain medicines, so as another option in pain therapy. So we'd use a high throughput screening process to maybe screen thousands of extracts through this type of screening process. So stepping forward to some examples of the sort of things that UQ has translated through this life cycle into an active agent, and it's particularly strong in the vaccine area. And you will have heard, no doubt, about Gardasil, and of course Professor Ian Fraser, and of course the massive advances in that area in terms of treatment of cervical cancer. And of course that vaccine keeps improving. I think it's now incorporating more agents in Gardasil 9. And of course, very topical, very recently, we know about the team led by Professor Paul Young that is also looking at vaccine research, and as part of a coalition of epidemic preparedness, the CEPI profile. And of course, they're having early successes in that space. There's such a quick start to the program where they've been able to achieve some positive outcomes in terms of antigen level in preclinical models already. So that's fantastic work that's going on there. And of course, that is part of that process through the drug life cycle. So here's another colleague of mine, and no, it's not Hugh Jackman or Kevin Rudd, but Professor Marie Smith, who's a pain researcher at UQ, received a Queen's Birthday 2019 Honours List to Companion Order of Australia. So only 12 of these are ordered. Hugh and Kevin got one, and of course Marie, who is my PhD supervisor. So just quickly looking at some of those, we've talked about the Gardasil already. We've talked about the new COVID vaccine, which is proving a wonderful outcome early. The Vaxxus also, the nanopatch system, which is a pain-free needle vaccination system. And you can see the sorts of dollars that come around with these sorts of investments. And then too, with Marie Smith, with the oral treatment for inflammatory neuropathic pain. So what typically happens with companies is in that drug lifecycle process, because of the amount of investment that has to occur, it's often sold on from a smaller company to a larger company in order to facilitate the very expensive clinical trials. And again, reiterating back on the costs, you can look at aspects of pharmaceutical industry, your medical devices, you can see the volume of sales there. And then of course, pharmaceuticals themselves, $1.2 trillion per annum, and then biopharmaceuticals as well. So it's a very large industry. In fact, in Australia, in 2018, it was reported that around 70,000 jobs are part of industry and part of research in the drug discovery or the MTP sector the medical technologies and pharmaceutical sector. So back to this life cycle process. And so now we have some insight. We've seen some parts of the discovery process here. We know that the COVID-19 vaccine is down in this phase. And then you can see very expensive phases in the clinical components as you build. There's even a phase 0 that exists in here now. And some of these clinical trial processes are combined. And there's also fast tracking that is applied to things like skin cancer and vaccines. They're run through separate committees. The essence of this project is managed by a government body, which is called the Therapeutic Goods Administration. That's the Australian form. You'll know it as the FDA in the US. So countries have their own forms. I guess the thesis behind the process here is actually that these organisations provide drugs that come through to market at this point that we know are registered medicines. They'll have an OSTAR number, and those medicines are proven for safety and efficacy. So that's probably the key outcome for the process and the role of the TGA. So, and then the 70,000 jobs I talked about are spread across this entire process. So we're focused a lot on the kind of role that I've got, which is down in research discovery and preclinical models. But then there's also roles around clinical trials, manufacturing, making sure that every tablet or every vial of vaccine that goes out there is the same as the last one that came through. That's an important part of that process. And then understanding the post-market surveillance and understanding safety and efficacy and reporting out there once those medicines are out in the wider population. That's very important, particularly for fast-tracked. So I've alluded to jobs, and this kind of starts our segue across to the MPIP. This is not an exhaustive list, and I'm sorry if there's anybody out there that's got a role in the MTP sector and you don't see your title up here, it's because there's only such a small font I can use. But you can see the diversity. Maybe these are roles that you never thought of before. The clinical research associates, project coordinators, roles in pharmacovigilance, that's around device safety and drug safety, regulatory affairs related roles, medical affairs roles. These are bridging out to the medical use of the products. Probably the traditional ones you'd thought about, manufacturing, packaging, but some of these you might not have considered before. Health economists, quality assurers. It's a whole myriad that fits into that 70,000 positions. So that's my segue. So now I want to talk briefly about where does the MPIP fit into this. It's a 32-unit masters, relatively new at UQ. It has also inside of it a graduate certificate as an 8-unit component, which is separate. You'll see both of these. So I'll talk a little bit about them, but both of these you can link through the Future Students webpage, which I've got a link at the back end so you can capture that and see that. But very quickly, I just want to talk a little bit about the program. So at 2 years full-time, it can be done in part-time mode. So the year one essentially covers that drug discovery life cycle. So understanding that span, understanding the roles, learning about the processes that are undertaken, the key important features of this drug life cycle as it passes and the device cycle as it passages through this process. So this is delivered both online and at PACE, which is where I am today, looking back across the river to UQ over my shoulder. Year two then, of course, is built around a couple of different aspects. There's either the possibility of doing 2 in-depth placements or one placement and a research project at this stage. So this is an independent research project which would occur at PACE. So that's so it's the integration of knowledge here, and it's the application with building more specific on-site knowledge. Of course, it starts in August this year with the COVID-affected calendar, and there's studies for 2021 look like starting in February. We all hope things going normal. So I guess in terms of what you're undertaking, it's a comprehensive suite of courses that exposes you to that life cycle management. So you're actually exposed to a lot of industry experts, a lot of world-class renowned researchers. Professor Ian Fraser, Professor Murray Smith, both came to present in courses last year as well, I could state. And we have a number of excellent industry partners that have helped build this program also come along and give their experiences and their insights or run tours out to those sites. So in essence, as well as building knowledge, all about knowledge forming, you're also developing skills, and those skills are important skills across a broad spectrum of those roles. So we're thinking about things like communication, leadership, project management. So being able to take initiative, being able to work teams is all important. So they're important aspects within this program. So you build on them not only in the first year together as a cohort, but also in your placement sites where that sort of initiative is expected actually. So, and then of course UQ has a range, and the school built into this program and around this program has a range of student support programs. So this is library skills, this is advanced ICTE roles in terms of advancing your communication and your written skills. Where are we undertaking this work? Well, this is where I am today, right now, at work. And this is the PACE, the Pharmacy Australia Centre of Excellence, which sits about a 20-minute walk across the river. Pleasant is a frame of mind, I guess. I've put that in there. It's pleasant now. In summer, it's not so pleasant, but luckily there's transport. We're situated next to the Prince Alexandra Hospital, which is a main hospital in Brisbane, and the bus line runs straight into UQ. So that's a reprieve in summer. Of course, another shot's probably of storm clouds coming across PACE. And of course, one thing I really love is the fact that we have a world-class library. UQ has a world-class library, and one of the library arms is out here in the PACE building. So it's a highly used resource. Then in our teaching spaces, this is one of our 3 teaching laboratories. We have a range of equipment which is industry standard. So this is both analytical and both pharmaceutical grade equipment. The other thing I'd like to note is you'll see these through the Future Students web pages. For international students, you'll see there's a possibility of applying for some of the limited scholarship, tuition-free scholarships. So that's between $5,000 and $10,000. So into the Future Students website, you'll see the link in there. For domestic students, I'd have to advise that at the moment we have a limited number of Commonwealth supported places. So these are 2 different processes for the 2 different types of cohorts looking at MPIP. And of course, there's the link to the Future Students site. If you have emails or inquiries, you can go through Future Students. Also welcome those to come in through the pharmacy link as well. You see the Facebook and Instagram pages there as well. And of course, welcome questions.
Hello. Thank you very much, Peter. I do have some questions.
Just getting
all. Yes, but a very active audience. So thank you very much for a very interesting talk. And the first question is about employability after doing this Master of Pharmaceutical Industry Practice, the MPIP. If a student came like from a bachelor's degree and then did this Master's straight afterwards, the question was, could they be employed in a pharmaceutical company? Do you think that's realistic?
I think it's I think it's a lot closer to the opportunity of being employed in a pharmaceutical company is a realistic opportunity. I think breaking into the sector can sometimes be tricky. It does require experience, And so that's why when we built this Masters, we had a large emphasis on this work integrated learning in the second year, so with the placement opportunities. And so for students who come into the program and really engage in that placement activity, I think you're opening up all of your possibilities to continuing on in the pharmaceutical industry sector. So I think there's a big advantage in engaging in that kind of activity, and I think that increases your employability massively with that. So it's unique in that we don't know of any other pharmaceutical sector engaging masters that has placement activities to this scope.
We know that, you know, having that experience while you're a student will really help you get a position, you know, after you graduate. So another question was about how is this program different from, say, studying advanced studies in molecular biology or something as specific as that?
Yeah, so that's right. So there are a number of masters around that have very specific scientific approaches. So a master's in molecular biology, you are going to do intensive amount of laboratory type work. You're going to learn a lot about molecular biology and the science around that. In this master's, you wouldn't do that. You're actually learning about the industry sector and you're learning about the drug life cycle process. It does mean that if you come in with a science degree, if you come in with some knowledge, you can apply that to that knowledge of sector, but you won't be undertaking any further science courses in terms of advancing your understanding of molecular biology. So they're quite different roles, and I think, and the courses therefore are very, very different.
Great. Well, we've got a very international audience. We've got another question here from about the placements in Year 2, and this person's coming from Europe.
Great.
And they're wondering if the placements in Year 2 are paid placements. So if the student receives any payment for those, and they're just not aware of the system in Australia.
Yeah, so that is a good question. And no, they're not paid placements. So the consequences of being paid, so the university takes insurance on the basis of your enrolment into the courses. So a paid placement would negate that insurance process. So so they're not paid. There are, of course, those scholarships. There are possibilities of bursaries being attached to some placements, but sorry, no, there's none that are paid.
That's fine. I have a question about COVID, the vaccine.
Oh, okay.
I don't know if you can answer this, but
no, probably
it's a very topical question. They're wondering what the next steps are in that process of developing the vaccine.
Oh, yeah. So that I probably know a little bit about. So for viruses and for treatment with vaccines for vaccine application, from what I understand, and this is purely from a peripheral view because I'm not at all involved in the project, but that team has had success in preclinical models. So that fitted into that part of that drug life cycle. The next stage for them is to accelerate quickly, if everything looks good, into the clinical trials situation, which is occurring with some other vaccines around the world. They go back in through the TGA, through a special committee that looks at viruses. And so the government is working with them in order to fast track this in as quickly as possible. Probably what's interesting is there are some vaccines, so say like the seasonal flu vaccine, because that's actually a registered vaccine, its approval process is even quicker because it's already been deemed safe many, many years. All they're doing is adapting it just for that new strain. So that actually even goes through a faster process where it doesn't have to undergo the rigorous testing. So, and across the world, which I know now that you're all out from different parts, every system's slightly different, but mostly at its core are these processes around safety and efficacy. Hopefully that helps.
Yes, That's great. It's good to know. It's on everyone's minds at the moment. You know,
Oh,
when
sure
will
is.
we get to a vaccine? I have a question about roles that you talked about. Someone could work in clinical trials, research roles specifically. Could you say a little bit more about what that work would be?
Yeah, so there's a number of placement opportunities, and probably post-COVID we'll have many more as well. The clinical research role is somebody that might be attached to a clinical research investigation unit. So exactly what we're talking about with the COVID going into clinical patient trials, the clinical research associate would sit in around that component, and they may have a role in managing and facilitating those clinical trial processes. So, and actually one of our colleagues has that role in Queensland Health, who's also part of the MPIP. So it's an integral role and really based around those clinical trials.
So a couple of other people have asked about the kind of background requirements, what degree they have to have in order to do this as a master's student. And was there, what if you don't have, for example, a science background? So what are the background requirements?
Yeah, it's, so obviously it sings to a science background, but there are a lot of degrees that have a science background built into them. So it's a matter of then us looking at what kind of experiences you have around chemistry or biology and those sorts of understandings that you've had in your undergraduate training. It's definitely, so the students that are currently on board, we've got students that have biomedical engineering degrees, we've got students that have pharmacy degrees, we've got students that have medical degrees. We have a whole range, as well as the applied sciences, the Bachelor of Sciences. So it's quite a diversity of students, and you all bring your own perspective and your background knowledge, which probably then also influences your perspective on where you might head in terms of the part of industry that you're looking at. So it's interesting to see that dynamic. But I think if you've got some underpinning science component, you don't have to just have a degree in science.
Excellent. So you kind of look at each application and the study that they've done.
Yeah, so that's basically how it works. And then there's some standard ones that just fit the bill right from the start.
Yeah. So this is a question about, I think, a comment that you made about the vaccine for diabetes. And the question was, is there still any use of pancreatic extract from dogs? Did I hear that correctly?
Yeah. Oh,
read that correctly?
So I guess dogs still they might get diabetes. Dogs do get fat, don't they? I think my dog never going to die. But so sorry, I can't help myself with dad jokes. I was told myself to not do that. So actually the extracts used to come from sources. So obviously it was a critical thing. They had a lot of people that were in diabetic comas, people very sick with insulin-dependent diabetes mellitus. This is not type 2, of course, which is a whole range of different medicines. So this insulin-dependent diabetes, the source was either bovine or porcine, so pigs and cows. And of course, if you could translate that into where we are now, that's a whole lot of pigs and cows giving up their pancreas. So we don't do that anymore. In terms of how that's done now, it's through cell systems. So much like the cell culture systems I'm running out in the lab now, there are cells that overexpress and actually manufacture insulin themselves. And we actually get that from that source. And these are human-based cells, so we get human insulin. Because the animal insulin, although it was effective, there were reactions because you're putting in a whole different species into your body. So people had adverse reactions to that. So Humulin is probably the one that, if there's any diabetics there, you'll know of that. And that's a human insulin injectable. So that's where we've got to in terms of insulin therapy. So not too far, but certainly no pigs and cows. No puppies are giving up their pancreas anymore. We've moved past that.
Excellent news. So I have a question about the packaging sector in pharmaceuticals. So how does knowledge in drug discovery and development help in that sector of pharmaceutical, the packaging sector?
So manufacturing and packaging, which might seem obvious, but they're under strict requirements and strict controls. So the information that's required to be on a package for a medical device or a drug is under strict regulatory control. So the industry has to comply with that. They'll have regulatory affairs control over that. So it's not as simple as what it might seem. And of course, the requirements are very, very specific, even on the sorts of things that have to be identified, what registered number they are, their batch number, their date. And a big part of that is tracking. So we know where they came from, when they were produced. And so the TGA, if they're doing a post-market surveillance on an adverse reaction, can track back to that package or that site, that day, that time, and maybe they'll find a whole batch, which has been the case in history. So it's really vital. The packaging itself has to be fit for purpose, and of course has to lower the risk of degradation, has to maybe ensure that there's sterility if the medicine requires that. So the packaging itself is in some ways an invention, often depending upon the type of medicine you're looking at. It might be an ampule, it might have to be opaque because the drug is light sensitive. So these are the sorts of things that are built in to the learnings in that part of the course of the program. So there's a course in the program that looks at manufacturing and packaging.
Oh, excellent. So there's a question from a more general question about the field. And the person is commenting that they've noticed a drastic drop of the number of compounds that make it to market. Are there reasons behind that?
So the drugs that get to market. So in Australia, I think the TGA is roughly approving about 40 new medicines a year. I think it's around about, which doesn't sound like a lot. The most important aspects, so in some areas, obviously it's got very competitive. You have to offer an advantage in order to have that drug come out into the market. Obviously, one is the industry earning its money. So if you don't offer advantage, nobody's going to buy it. So then that's pretty much done and dusted. And the other is an improvement in efficacy, which is obviously something that society wants. A better option, lower side effects. So in some areas that's particularly challenging. You think of areas such as antimicrobials, antibiotics. That's a very challenging area, which has probably been underinvested for a long time. The trends that will be followed obviously are driven by the economics of the scope. So the larger the impact industry can have, so the broader of the scope of application of a medicine, the better are its chances of actually making a return. I know that might sound deleterious, but if industry doesn't make a return, there isn't any business and we don't get new medicines. So you have to have, they have to make money in order to I guess, build new medicines. But that's probably the challenges. I think it will, the new medicines, I think, will continue with a steady flow. I think it's probably in different areas that we see that the space becomes very narrow. And so the opportunities for a new medicine in certain spaces are difficult. Obviously, the booming area at the moment is biologicals. There's a lot of new biologicals out there. And so these are like the anti-TNF alphas and all those sorts of compounds because they're so useful in treating things like cancer, big diseases. So hopefully that helps.
Yeah, and another question about whether the course covers areas of reimbursement policy and HTA. I'm not sure what that is, or only regulation.
So I'm not a specialist in that, as they will know, and I clearly know, and if they know those aspects, they know that. So yes, it does. So we have, so we're a School of Pharmacy here, and of course our engagement is into the pharmaceutical industry. And so we have experts across a whole range of different fields. And one of those happens to be pharmacovigilance and pharmacoeconomics, and that's where those HTA processes fit in. So funny enough, we were only talking about that last week. So yes, there are aspects covered in that. Obviously, the financial viability of a product is core to it actually being present and viable.
Excellent. There's another question about the program, the qualification at the end, and is it the person doesn't become an Australian industrial pharmacist? That's the question. Is there a particular title connected with successful graduation of this program, or any registration or accreditation?
Yeah, no, there's no registration or accreditation. It's a non-accredited program. So in Australia, the pharmacy term is a restricted term that applies only to the registrable pharmacists. So all pharmaceutical science programs in Australia, so there are a number of Master of Pharmaceutical Science programs, they're not registrable programs. If they use the term pharmacy, then they relate to that registered accredited body. So it's very, very separate, and so the name is very separate. So you could, so the Master of Pharmaceutical Industry Practice, you could by name think of it as that of an industrial pharmaceutical scientist, is probably how I'd term it if you were trying to define yourself outside of the degree name. And that really is what you would become, except we've tried to engineer this, well, we have engineered this, that it's so much more, because it also points into pharmacovigilance and also points into health economics and those sorts of components, registration, medical affairs, which a traditional pharmaceutical sciences masters would not address. You'd probably find that you're focused in those very much at things like drug formulation and manufacturing packaging.
I think that's a great spot to end there, Pete. I think you've given us many ideas about all the different opportunities that come from it. And I think the important thing here is the support that this program has from industry, not necessarily just strictly whether you can register or not as a pharmacist, but you've been working very closely with the pharmaceutical industry in developing this program. So I think everyone agrees it's growing in popularity and it has enormous potential. So we do have a number of quite specific questions that have come up in the chat, and I think we'll provide those questions to the School of Pharmacy. They're questions about individuals' eligibility for the program. Cool. And I think they're best answered rather to that individual person rather than to the whole group. But thank you all very much for coming today, and there will be a recording, there has been a recording made of this, and you'll be sent around a link after the presentation today. But I'm sure you want to join me in thanking Pete Cabot for his presentation, and thank you all very much for coming.